Imagine a factory that doesn't use massive smokestacks, chemical vats, or heavy machinery. Instead, this entire factory fits on the tip of your finger, and its workers are billions of microscopic organisms. Yeast. This is the power of synthetic biology. We're taking baker's yeast, the same simple microbe used to bake bread and brew beer, and rewriting its genetic code to build the future of manufacturing. Yeast cells run on DNA, which acts like their operating system. By editing this biological software, scientists can force yeast to consume simple sugars and churn out complex high-value molecules on demand. Instead of producing alcohol, these reprogrammed yeast cells become cell factories. They can manufacture everything from spider silk and clean jet fuel to life-saving human medicines, completely bypassing traditional agriculture. To start, scientists must insert new instructions into the yeast. They often use plasmids, which are floating loops of DNA. While easy to insert, plasmids can unfortunately be lost over generations. For a permanent change, scientists use genomic integration. This stitches the foreign genes directly into the yeast's chromosomes, ensuring the new instructions are passed down to every future cell generation. The ultimate tool for this rewrite is CRISPR-Cas9. This molecular search and replace tool cuts specific DNA sequences, allowing researchers to easily knock out unwanted genes and paste in custom target instructions. To control the process, scientists use promoters. These act as genetic volume knobs, telling the yeast when and how loudly to express the new genes, often activating only when specific sugars are present. They also act terminators, which serve as genetic punctuation marks. This tells the cellular machinery exactly where a recipe ends, preventing the yeast from misreading the code and wasting energy. Once the software is installed, scientists rewire the yeast's metabolic pathways, which are like cellular plumbing. They shut down the default pathways that make ethanol to keep sugar from being wasted. They redirect this carbon flow toward the target molecule instead. They must balance the cell's energy and cofactors to prevent bottlenecks that could choke or kill the yeast. Consider human insulin. Yeast is engineered with the human proinsulin gene. Because yeast cannot naturally process the 2-chain hormone, it secretes a precursor molecule that scientists collect and fold. This yeast-based insulin costs just $4, 28 to us it's $16 per vial to manufacture, yet complex supply chains and patent markups have historically driven U.S. retail prices over $300, $0 per vial. Vanillin is another prime target. Natural vanilla beans are labor-intensive, driving costs between $200 and $4 per kilogram due to volatile crops and hand pollination. Petrochemical vanillin is cheap, costing only $0 to $20 per kilogram. However, because it comes from fossil fuels, it cannot be labeled as a natural flavor. Yeast fermentation solves this. It produces bio-vanillin for $100, $0 to $300, $0 per kilogram. This qualifies as natural, offers stable pricing, and uses 0 petrochemicals. But vanillin is toxic to yeast. To survive, the engineered yeast attaches a sugar molecule to the vanillin, making it harmless. Scientists simply snip the sugar shield off during purification. Scientists are also engineering yeast to make spider silk. However, the genetic code for spider silk is highly repetitive, making it difficult for the yeast to read and build. Recombinant silk proteins cost $35, $0 to $50, $0 per kilogram to make today. Engineers are optimizing codons to drop costs to $20, $0 to $30, $0 per kilogram to compete with nylon. For biofuels, yeast is engineered to produce farnesene, a hydrocarbon molecule. This is easily converted into farnesane, a clean drop-in replacement for diesel and aviation jet fuel. Currently, farnesene costs $4, $0 to $5, $0 per liter to produce. Engineers are working to hit a target of $0 per liter to make it commercially competitive. To produce these molecules at scale, scientists moved the engineered yeast from small petri dishes into giant stainless steel bioreactor vats holding up to 250 liters. These bioreactors act like luxury cruise ships for the yeast. Operators must maintain a perfect thermic temperature, regulate pH, and pump in oxygen to keep the cells healthy. Glucose is added using a fed batch method. By feeding the yeast slowly, scientists prevent the cells from producing toxic amounts of ethanol, keeping them in peak production mode. Once fermentation is complete, the mixture undergoes downstream processing. High-speed centrifuges and filters separate the yeast cells from the nutrient broth containing the target compound. If the compound is kept inside the cells, the yeast walls must be lysed or burst open, using high pressure or specialized enzymes to harvest the product. If the yeast secretes the product, it is extracted directly from the liquid broth and chemically purified into a market-ready, high-purity compound. This biological manufacturing offers a sustainable path forward. It reduces our reliance on petrochemicals and intensive land-hungry agriculture, using living software to build a cleaner world. From medicine to materials, reprogrammed yeast shows that the future of manufacturing isn't built in traditional factories, but grown inside living cells. By turning biology into technology, we can manufacture a cleaner, healthier future. Subscribe for more deep dives into the science shaping our tomorrow. Layman's field knowledge for everyone.